who first observed Plasmodium in human blood? Who first observed Plasmodium in human blood? Who first observed Plasmodium in human blood? Can anyone tell me? Who first observed Plasmodium in human blood? Anyone? Raise your hand. Okay. Answer is Leveran. Remember this thing. But he first observed it. Then comes Ronald Ross. Is the vector of Plasmodium. Before that, there was no idea how Plasmodium comes to human blood. And after that only, the mosquito eradication program started. Before that, there was no such idea how actually the Plasmodium is coming into the human blood. And for this revolutionary discovery, Ronald Ross got Nobel Prize. So this is the most important thing. And how did he discover this? He first observed some stages of mosquito in the stomach of female Anopheles mosquito. By dissecting the stomach of female Anopheles mosquito, he first observed that Plasmodium is actually carried by the female Anopheles mosquito. Now, the most important question I am going to ask now, it is regarding the two terms. I think I discussed or not, I forget. Vector and carrier. Now, Orko Prabhu, do you hear me?
Yes, sir.
You tell me the difference between vector and carrier.
Sir, vectors carry a pathogen without.
Tell me.
Sir, vectors carry a pathogen without getting infected themselves.
Okay, carrier.
And carrier, sir, I'm not recalling the carrier.
What? I'm not getting you.
Sir, I'm not recalling the carrier.
Okay. Thank you. Anyone else can say? Anyone else? Okay. Look one thing. You know, I am giving two examples. One is a mosquito, those female mosquito. Other is housefly. Two different examples. One is mosquito and the other is housefly. Now you see, whenever the term mosquito comes, mosquito is carrying Plasmodium. Housefly is also carrying some pathogens causing typhoid, cholera, etc. Now, both are carriers. As carrying, housefly is just mechanically carrying the pathogen from one food to the other food, and in this way it helps in causing the disease. Mechanically carrying. Housefly is mechanically carrying. Now, what about the female mosquito? While female mosquito is carrying Plasmodium, it is found that some stages of life cycle of Plasmodium are found to occur in the body of mosquito. So in order to be considered as vector, wherever you can call an animal vector, if some stages of the life cycle of the pathogen are found to occur in the body of that. So vector is a carrier inside the body of which some stages of life cycle of Plasmodium or some stages of life cycle of the pathogen are found to occur. Is it clear now? So what should I say? Vector is a carrier inside the body of which some stages of life cycle of plasmodium of pathogen so the body of which the body inside the body of some stages of life cycles the pathogen are found to occur. So if a carrier can only be considered as vector whenever some stage of the pathogen it is carrying are found to occur inside the body of which. And which is just a carrier, not a vector, which is just mechanically carrying the pathogen. So it is a housefly only carrier, but not vector. Why? As it mechanically carries the pathogen. So vector is also a carrier. But the difference between them is, in case of housefly, it carries mechanically. No stage of life cycle of pathogen is found to occur inside the body of housefly. So it cannot be considered as a vector. But female mosquito is a vector. Why? Because some stage of life cycle of Plasmodium are found to occur inside the body of female Anopheles mosquito. I think you can remember at gametocyte stage, Plasmodium enters the body of female mosquito, and then after that you see it will form the male and female gametes in the body of mosquito. Then in the stomach of the mosquito, fertilization takes place, zygote is formed, comes out of the stomach, forming okinete, which in turn will form the oocyst. Oocyst then undergoes sporulation to form the sporozoites. So all these stages, gametocyte to sporozoites, found to occur inside the body of female mosquito. That's why it is considered as what? The vector. That's why female mosquito is considered as a vector. Another question I am going to avoid ask. Do you hear me?
Yes, sir.
Now you tell me, malaria patients are found to shiver. Malaria patients are found to shiver. You just tell me, why do malaria patients shiver?
So can you please repeat?
It is found that malaria patients shiver.
Yes, sir.
Now the question is, why do malaria patients shiver?
Sir, malaria patients shivers because the hemozoin toxin it does a exothermic reaction which produces heat. The heat receptor through the sense means by the sensory impulse it goes to
Neuron, the impulse goes to muscle.
yes sir. Contraction starts and the shivering occurs and finally fever comes.
Why after shivering fever comes? Okay, over next anyone else?
Of the body rises.
Actually, due to muscular contraction, there is the production of heat, energy, ATP.
Yes, sir.
And so that the temperature is reached.
Of the body rises and that's fever comes.
And fever comes. Okay. Now, I am coming to the next group. Locomotory organ, cilia. And in this case of paramecium, cilia are found to be distributed throughout the body surface. Nutrition, holozoic mainly. There are some parasitic form. In their case, it may be parasitic nutrition. Next, asexual reproduction, mainly most important is by, remember this thing, transverse, binary fission. Asexual reproduction, you need to remember this one by transverse, binary fission. This is carried out by transverse binary fission. Then you see in case of other things are presence of two nuclei. Macronucleus, micronucleus. It is also called vegetative nucleus because it controls metabolic activities of the cell. Micronucleus, it takes part in reproduction. Macronucleus means vegetative nucleus, micronucleus, it takes part in reproduction. It controls the metabolic activities of the cell. Presence of contractile vacuole. You know, helps in osmoregulation. See, if you go to the diagram, so what happens here? So you see here, These are the two contractile vacuole, two in number. Secondly, you will get the macro and micronucleus. You see, macronucleus you get here, and so this is the and micronucleus. Here you will get so cilia is found to be distributed throughout the body. Cilia is found to be distributed throughout the body, and here you will get the buccal cavity. So having two contractile vacuole and some other things are found here throughout the body. Paramecium, example is paramecium. And it is also called slipper animal. Why? Because it looks like slipper. Someone trying to enter by Galaxy J4. Please, on behalf of me, please, anyone can tell me in the group that you will not be allowed by Galaxy J4. Now you see, second thing, nutrition, it is, paramecium is microphagial. It is called microphagial. Why it is called microphagial? It is called microphagial because it feeds on bacteria. It is called microphagial because it feeds on bacteria. Macrophagial. The nutrition is microphagial, means it feeds on bacteria. It feeds on bacteria. You will get some food particles here and there. Now, regarding sexual reproduction, it is mainly conjugation. Regarding sexual reproduction, it is conjugation. That is here, a conjugating tube is required to form between two paramecium to facilitate sexual reproduction. Now, paramecium, the spelling earlier it was like this, nowadays spelling appears like this. So you can write these also. Now next comes to the is specific particle present in it called kappa particle. Kappa particle is found in some strain of paramecium. That means depending on the presence of absence of kappa particles, two types of paramecium, killer strain, sensitive strain. Possess devoid of kappa particle. Kappa particle secretes remember this thing. Kappa particle secretes paramecin toxin, and whenever sensitive strain of paramecium are found to be in contact with the kappa particle, that is paramecin toxin, they will die. So sensitive strain of paramecium, they get killed if comes in contact with the, if they come in contact with the paramecin toxin. So the killer strain is with kappa particle posing paramecin toxin. Sensitive strain is devoid of kappa particle. So this is what about the two strain of paramecium. So ciliata completed. That is, this is the end of kingdom protista. So what I call kingdom protista. In class 12 you will get it. This is the inheritance of kappa particles. I'm writing here. Inheritance of kappa particle. It is actually cytoplasmic inheritance. Inheritance of kappa particle is nothing but the cytoplasmic inheritance. So we are now proceeding towards the next kingdom, kingdom fungi.
Sir,
Yes,
so cytoplasmic inheritance means
actually you will get it later on actually. When you need to study inheritance, basically there are two types of inheritance. Some characters are actually carried from, from cell to cell or organism to organism or parent to the daughter organism through cytoplasm. We have also some cytoplasmic inheritance also in our case in human being also. So in every organism there are some inheritance carried by cytoplasm, like mitochondrial inheritance. Okay, so this is called, actually here nucleus is not going to take part, but the cytoplasmic DNA helps in the actually inheritance of that characteristics. There's such a type is called cytoplasmic inheritance. Are you getting me now?
Yes, sir.
Now we are proceeding towards the next kingdom. Kingdom fungi. All it includes all multicellular eukaryotic heterotrophic organisms with cell wall. So what are kingdom fungi? You need to remember these things. First of all, multicellular, eukaryotic, heterotrophic, and with cell wall. Most important, with cell wall. And if you go to the kingdom fungi, the most important characteristics of fungi, they are the plant group, the one of the kingdom of 5 kingdom classification. They are reproducing mainly by forming different types of spores, mainly by forming or producing different types of spores. Then you can call it actually the kingdom fungi. Now I am to classify this kingdom first. Then one by one I am to discuss. Phycomycetes, Ascomycetes, Basidiomycetes, Deuteromycetes. So kingdom fungi include the following: Phycomycetes, Ascomycetes, Basidiomycetes, and Deuteromycetes. So here among this group is the most interesting group. Among them, Deuteromycetes is the most interesting group. Why it is called so? Why it is called most interesting group? You see, in Phyco, in Asco, even in Basidiomycetes, you will get both sexual and asexual reproduction. That means the organisms under Phycomycetes, organisms under Ascomycetes, organism under Basidium. I am giving some examples. Suppose, then it will be easier for you. Your example, suppose it is a myopod. Ascomycetes, suppose Ascobolus. Basidiomycetes agaricus, which is called mushroom. And Deuteromycetes, suppose Helminthosporium. Now you see, if you go to life cycle of myocod, if you will go to the life cycle of Ascobolus, go to agaricus, that is mushroom, you will get both sexual and asexual phase. They can reproduce by both sexually and asexually. But when comes to the deuteromycetes, most important thing, remember, sexual phase is found absent. Sexual phase is found absent in deuteromycetes, and that's why you can call it again what? Fungi imperfecti or imperfect fungi. Why imperfect fungi? Because sexual phase is found in this group. Now suppose if any fungi, earlier it is called suppose Phycoma under deuteromycetes, but after some time or somehow in some organisms of deuteromycetes, if sexual phase is found, then what to do? Are you getting me? If sexual, I will be sending the ideal password again, don't worry. So what happens if somehow sexual phase is found, then what to do with this organism? Is it still under deuteromycetes or there should be any change in it? Remember this thing, if in any organism which is normally under deuteromycetes, if somehow sexual phase is found in it, then it is to be excluded from deuteromycetes and renamed so it's a fungal cell, and the outermost you can call it the cell wall made of mainly made of chitin. Inner to which you will get the cell membrane, nucleus, you will get the mitochondria, ribosomes. So this is what about the structure normally, and some other fat globules are found here and there. So most important thing you need to remember here that the fungal cell wall is made of chitin. Can any one of you tell me what is chitin? Chemically, what is chitin? Anyone please?
Sir, the
Right.
नाइट्रोजन containing nitrogen containing polysaccharide fungal cell wall আছে
It's a nitrogen containing polysaccharide. Chitin. Besides chitin, it is also called other name of which is Fungal cellulose. Now in fungal cell, an invaginated structure is found in the cell membrane. A part of the cell membrane gets invaginated to form a structure like this. And you know this invaginated structure is called, what to write? Lomazome. So cell membrane invaginated inside to form a structure in fungal cell which is called lomazome. Basically it is concerned with the production of spore. So this is what about the basic structure of the fungal cell. You will get the cell membrane. Similar, it is made of protein, lipid and protein. And other thing remains same. Nucleus is a true nucleus. Mitochondria are there. So you need to understand one thing here. One is the chitin, the fungal cellulose. Lomazome is found here. So this is what about the fungal cell. Now we are coming to two most important terms in the fungi, which is
Sir.
hypha.
Sir.
Yes. So,
Sir, please explain the lomazome.
lomazome actually is cell membrane. Cell membrane, you know, cell membrane invaginated, enfolded inside to form a structure called lomazome. You can call a membranous vesicle. You can call it also, lomazome is a membranous vesicle. And it is concerned with the spore production during asexual reproduction.
Sir,
Yes?
you were talking about deuteromycetes.
Oh, yes, okay. I forget it. Sorry, extremely sorry. Okay, I'll be coming to that. Now, is it clear, Oditro?
Yes, sir.
Now I am coming to one by one. Okay, I was talking about deuteromycetes. Yes. What I said, that in deuteromycetes, sexual phase is found absent. If somehow sexual phase is found present, then what will happen? Then this organism, which is initially under deuteromycetes, it is required to be removed out from this group. So if sexual phase is found in any deuteromycetial fungi, first thing to do, this fungus is required to be removed out from this group. It should be no more under deuteromycetes. Then it is to be renamed. Earlier name should be changed and new name is required to be given. Then its characteristics are required to be observed. If the characteristics of these fungi are found closely related to Ascomycetes, then it is to be grouped under Ascomycetes. If it is related to Basidiomycetes, then it is to be grouped under Basidiomycetes. Generally, deuteromycetial fungi are having the connection between Asco and Basidio actually, not Fico. So, either these deuteromycetial fungi after the discovery of sexual phase to be included under Ascomycetes or under Basidiomycetes depending on its similarity with the group. This is one about the deuteromycetes that I was discussing. Clear? Then comes to the each group. I am to start with each group now.
Sir.
Yes.
Sir, can spore formation be sexual and asexual both?
Asexual. Spore formation cannot be considered as until actually gametes are formed. You know that spore formations are in higher plants. Initially microspores and megaspores are formed. Up to spore you cannot call them gametes. But yes, in some cases spore formation takes place by meiotic division. But in lower classes, spore formation takes place by mitotic division. In flowering plants, even in bryophytes, in pteridophytes, and in spermatophytes. মানে except algae and fungi, here spore formation occurs through meiosis division. These spores, basically up to this, it may be asexual structure. But you know, as the spores are to form gametes, they may be involved in the sexual structure. So, spores may take part in asexual reproduction. It may again take part in sexual reproduction in higher plants like bryophytes, pteridophytes, and what else they said? Spermatophytes. Do you know what are spermatophytes?
Seed producing plants.
Who are they? One is two groups are there.
Phanerogames. Gymnosperms and gymnosperms and angiosperms.
Very good. They are the gymnosperms and angiosperms. And in these, spores may will be producing gametes. Spore will be in turn producing gametes. From spores, gametes are formed. But in lower group, spores are truly in asexual mood. In higher plants, spores may take part in the sexual reproduction beyond by producing gametes. Is it clear now? Who asked me the question?
Yes, sir. Sir, then in Deuteromycetes, there is chromosome.
Basically, you know, spores are formed. You know, in case of mycetes, mute karo. Rajshekhar Dev, mute karo. Now you see, in case of Deuteromycetes, they are reproducing by conidius. I will be giving you later on. They are reproducing by conidia. So here, chromosome may not be required. Or even if it is present, this is not create any problem. Because here, spores are not going to produce gamete in fungi and algae. Are you getting me?
Yes, sir.
Okay. Now, because spores are growing further to form male gametophytic body. Suppose in bryophyte, in pteridophyte, if you see the spores are to produce the male gametophytic body or female gametophytic body in from bryophyte to phanerogames. But in lower group level in algae and fungi, spores are truly for asexual structure. Spores are not the gametes. Another thing you should know, the spores are not the gametes. So you cannot say spores are sexual structure again. Are you getting me now?
Yes, sir.
Okay. Now next comes the first group. Kingdom fungi. Kingdom fungi, then first group is Phycomycetes, the simplest group. Basically, septum formation. Depending on the septum formation, you can classify fungi into two groups, accepted fungi and the septate fungi. Phycomycetes is actually accepted, and septate includes Ascomycetes and Basidiomycetes. Although septa also found in Deuteromycetes, so you can, they are also septate.
Sir, video is unstable.
Which one? For video unstable? Don't think so. Video unstable? If I am depending.
Sexual reproduction is not found in
Deuteromyces.
sexual reproduction
The
is
bo-
found
bo- bo-
in
Then
আঙ্গিরে দুইটা ভাগে ভাগ করছে দেখে কথা
accepted
এটার আবার ভাগ করছে
phygomyces.
are separate gitu
separate
Septate.
are separate
You will get
separate
gastromycetes.
দুইটা ভাগ করছে আঙ্গিরে
separate মানে কি
And
দুইটা আবার ভাগ করছে separate দেখো সেকেন্ডারি
তুই কই যাস
I will be telling you, it doesn't mean that Deuteromyces is accepted. No. Deuteromyces is actually separate. But these are confusion would sometimes come. So, these are the basic confusion. Now I have to start with the phygomyces. Complete quickly. Complete quickly. Because Deuteromyces are fragile and also soft. That's why in this confusion, you understand that you get the Deuteromyces. Here, now phygomyces. I am discussing with an example like neopol. Look, and you know what it is called? Fungal mycelium. Each structure you can call it aerial hypha. You can call it Takara hypha. So that hypha, can any of you tell me what is hypha? You got it in lower classes. Either in 7 or 8, you got it. What is hypha? Hypha. Structure producing fungal body. Hypha or hyphae are slender, thread-like structures producing fungal body. That is, you can call it mycelia. A number of hyphae together form mycelium. So we can say hypha is the unit of mycelium. As well, the fungal body is called mycelium, and each unit, each structures forming this body or mycelium called hypha. Now, in case of neopol, we are getting two types of one is aerial hypha, which grows around the soil, and there is fungal hypha, by which it derives nutrition from dead and decaying organism. Generally, in neopol, most of the cases it is saprophytic fungus. It is saprophytic fungus. Is it clear now? So you see, the next what to do? Now whatever is structure, first of all, accepted and multinucleate. You need to remember this thing. What we call it in Phacomycetes, the mycelium is accepted and multinucleate. And you know, multinucleate mycelium is called Sinocytic mycelium. The multinucleate mycelium is called sinocytic. The term used with the multinucleate condition. Sinocytic mycelium actually has septa is not developed. You see no septa is found. That's why sinocytic condition can be developed easily. Once septa developed, you will not get the sinocytic condition because you will get the nucleus in one cell then. This is the multinucleate because septa have not developed. Now here it is reproduction. First of all, asexual reproduction by sporangiospores. To the tip of the aerial hyphae, a small solan structure is developed initially. Later on, the enlarge in size. From the mycelium, cytoplasm and nucleus is carried towards the periphery. Next diagram. Enlarged in size. And cytoplasm is nuclear. Partially arranged towards the periphery of the sporangium. And in the center, you will get a vacuole. Vacuoles are arranged in the center. So, this peripheral region is called poroplasm. And vacuolated region is called, to write. Columella. After that,
Sir,
yes,
sir, which part is poroplasm?
peripheral red dotted plant is actually the cytoplasm and nucleus. Wherever actually the sporangium is formed, cytoplasm and nucleus are from the hyphae carried to the periphery of the sporangium. And this red dotted part indicates the sporoplasm and bluish part indicates the corpuscleplasm. Who are they? They are only the vacuoles. Vacuoles are found to be arranged in the center. The next diagram now you see
Why, man?
in the periphery, the nucleus in cytoplasm aggregate to form, they are now called sporangiosperms. And vacuoles aggregated to form a structure, and it is called columella. Columella not going to take part in the, now this structure you know it is called sporangium. With the stop you can call it sporangiophores. Sporangiophore, what is stop? When it matures, one of the sporangium is broken down. When the sporangium is broken down, and spores are divided out. These spores on germination give rise to neomycelium. This is how muco reproduces asexually by means of unforming sporangiospores. Next one. So it's very clear now, I think it's clear. Sporangiospores. Yes, tell me. Follow? Right. Format, most important is coming in the exam sometime. This is called colonal stage of mucosa. What is this? Basically, so it is concentrated sugar solution. And those underlying is placed here. Then what is found after that? This is actually broken down into normal segments. Hyphae is broken down into number of round or oval shaped segments. Each are they are called oidia. Visually, oidia gets detached each oidia will give rise to neomycelium. So if mucus is placed in highly concentrated sugar solution, function, the fungal algae is broken down into a number of oval or round sheet segments and they are called oidia or detachments. When they get detached from each other, they will give rise to new mycelium. Another type is found sometimes, which is called chlamydospores. Igual are sometimes found in the hydra. So this is the hydra. This is a hydra, suppose, and you are getting some equal spores here. There are equal spores. Formed under unfavorable conditions. So these thigmospores are called chlamydospores. These thigmospores are called chlamydospores and they are formed under unfavorable conditions to provide protection. When favorable condition comes, these thigma structures get dissolved and they will give rise to new mycelium. They will give rise to new mycelium. This is what about the chlamydospores. Is it clear up to this? Myocor. In myocor generally we get the sporangiosperms mainly. Then after that we may get what is called the solar stage where olia is found. And in some cases motile spores are also occur. Suppose motile spores or juice spores are found in Phytophthora. Motile spores, or remember this is the most important one. Motile spores are called juice spores are found in where? Phytophthora. Motile spores appear like this. They are the motile spores. They are the juice spores, vegetative spores. And they are found in Phytophthora. I think you know what is Phytophthora. Phytophthora, have you ever heard of a disease? It's called leaf blight of potato. Leaf blight of potato caused by Phytophthora infestas. Caused by leaf blight of potato is caused by Phytophthora infestas. This Phytophthora will get the motile spores. So I have to stop here today. Next Thursday, yes. Problem is, I will be explaining later next day that if it is grown in the highly concentrated sugar solution, mycelium is broken down into a number of pieces. Each one is called oidia, and they will give rise to new organism. Okay, so I will be in the next class.